Hello all, welcome back to the BioRocks channel. Today in this video, we will be learning about the transportation of molecules across the plasma membrane, that is your cell membrane, which is partially permeable to few of the molecules or the ions. So we'll be seeing the processes like diffusion, facilitated diffusion, as well as the active transport in this video today. So what is simple diffusion? Simple diffusion is going to be something which speaks about the movement of the particles, molecules, or ions from a higher concentration towards a lower concentration. If you see here, you can see that the intracellular, that is inside the cell, the concentration of this particular molecule is very, very less when comparatively to that of this particular concentration outside the cell. So because of the kinetic energy of these molecules, they are going to move from the higher concentration towards the lower concentration until and unless they are going to build up a equilibrium, that is maintaining the same concentrations outside as well as inside the cell membrane. Okay, so they are now in an equilibrium. So if there is high concentration, then what happens is, in order to maintain the equilibrium, the molecules will be using their kinetic energy to move from a higher concentration to the lower concentration, which we call this process as your simple diffusion process. Okay, so remember that this, what you're seeing here, is going to be your cell membrane, the bilipid layer that we are going to have. We know that all your lipids, whichever are going to be the ones which are non-polar, while if you're going to look at your what we call as the water, which is going to be your polar molecule, which is going to have the H plus and the OH minus, that is O minus if you're going to see. So we are going to call it as polar. Let me show you, children, what are these polar and non-polar, such that the concept will be clear for you all, whoever are listening to this concept for the first time regarding the diffusion process, children. So remember that the bilipid layer. So this is going to be your plasma membrane, which is a bilipid layer. Okay, so this is going to be a bilipid layer. So we all know that lipid is nothing but fat, right? And your fat is made of carbon, hydrogen, oxygen. But this particular fat does not have any poles. It does not have any poles, no poles at all. Poles in the sense, no plus charges nor no minus charges. Okay, so it's going to be called as a non-polar molecule. Fat is a non-polar molecule. Okay, so to move something from the bilipid layer, they have to be what we call is soluble. They should be the molecules. Let me write this for you. Molecules have to be soluble in lipids, to move across the cell membrane, This is a rule because these are non-polar. So non-polar molecules which are soluble here can only move across the bilipid layer. But we know that in a bilipid layer, there are even some integral membrane proteins which are being present. So these are the proteins which are being present. For instance, if you want few of the things to move across now through your membrane, but if that is going to be polar, then how will it be possible for them to move? So that is also one condition. So I'm telling you that the plasma membrane is a bilipid layer, but interrupted with some of the membrane proteins. They are having some membrane proteins. They are being interrupted. And these membrane proteins, children, what they are going to have is, they are going to have a channel in between, which is going to be filled up with the water. Okay, which will be filled up with the water, children. So this is the one. So now what happens is, all the water soluble ions, whichever are being present, can easily now move from this particular thing, because here it is completely a lipid layer, and lipid layer is going to be non-polar. So it will not allow the water soluble things to get along. So, but now what has happened in the membrane proteins, you're having a channel where you have water filled up. So it will easily allow the water soluble things to move across, right? So the water soluble molecules can move easily across from outside to that of inside. So these can move. So this is a possibility by using the membrane proteins. So we can even call these membrane proteins as carrier proteins. We can call them as carrier proteins, or you can otherwise call them as a transfer proteins also. Transfer proteins. We will see them one by one, but just remember that your bilipid layer is non-polar. To move from a non-polar things, only non-polar things can only move, which are soluble in the lipids can only move across. But to move the water soluble molecules, we have this specially inbuilt what we call as a transfer proteins which are being present. I hope now you are understanding. If you look at the water, children, let me make this out. So water is going to have a bent structure like this, and your O is going to be electronegative, and slightly your hydrogens are going to be positivity. That is, they are electropositive, and the oxygen is going to be electronegative. So we call our water molecule as polar molecule. Polar molecule, because it is bearing two types of charges here. One is negative charge, the other is going to be positive charge. So we call them as polar. But our fats are neutral. They do not have any poles, so they are non-polar. I hope now I made it clear for you, children, that why is this basic difference coming up there. Okay, so now I'm speaking about the simple diffusion process, children. So we are making a movement from the higher concentration of molecules to the lower concentration of molecules. We are not investing any ATP. It is just the kinetic energy of the molecules which is being used to move from higher to the lower. And we all know that the simple diffusion is going to be possible where it's going to be possible only in the fluids as well as the gases. So this point is important for you to remember, that simple diffusion, the molecules are using what? Molecules use their kinetic energy. Okay, they are not using, no ATP is used. No ATP is used, dear children. This is also important. Okay, this can be seen because molecules having kinetic energy that you can see only in the fluids as well as and you can see in your gases. So simple diffusion is a process that is going to happen in the fluids as well as gases, but not your solids, children. So this is one of the processes, that is simple diffusion, by which the molecules can move across the cell membrane. Another process that we are going to have is your facilitated diffusion. This is your facilitated diffusion, children. So what is facilitated diffusion here is nothing but this is your facilitated diffusion. Facilitated diffusion, because diffusion is movement of ions from a higher concentration to lower concentration. So we already are seeing that here the molecular concentrations are more, while the molecule concentration is less here. But what is happening is, children, the molecule sizes are big. Okay, the molecule sizes are big here. The molecule sizes are big. But when you see, when you talk about the diffusion process, in the diffusion, the molecule, so let me even write that, children, and make it clear for you that what is a basic difference here now, that diffusion and facilitated diffusion is going to be that your what we call as the diffusion process only what is happening is, in the diffusion process, only small molecules are being moving from or across the semi-permeable membrane. Okay, but in facilitated diffusion, we see that we are moving the large molecules. Like example I can give you is, we are moving glucose or we can even move amino acids here through the facilitated diffusion process, children. And you can see that this particular transport protein, which is present here, as I told you, is going to have a channel in between, which is going to be filled with water, due to which the water soluble molecules can easily be moved across. Water soluble ions can be easily moved across, that you're seeing here. And another thing that you can see here is these carrier proteins that you can see. Once they are going to be fixed with a complementary molecule here, their shape is going to change. See how it's been changed. Their shape is going to be changed. And once if their shape is being changed, now what happens is the molecule is going to be led to be free flown into this particular, the other side of the membrane. So you can see that the carrier protein is going to bind with a complementary molecule, due to which the binding process, what happens is due to the binding, the change, there is going to be change in the shape of the carrier protein, which enables it to open towards the other side. So it's like a flip, right? It's opening on other side, but it's binded. The shape is being changed. It's opening towards the other end, and the molecule is going to be thrown into the other side. So this is how the carrier proteins, or the transfer, what we call as transport proteins or transfer proteins, which are being interrupting the cell membrane bilipid layer, are helping in the movement of the molecules which are having the larger sizes from a higher concentration to lower concentration. And remember, children, here also there are no usages of ATPs. In the facilitated diffusion also, there is no use of ATP molecules. This is another thing that you can remember, okay? And the next thing that I would like to show you is, children, regarding the next one that we have is going to be another procedure which is being called as your active transport. It's going to be the active transport. So in the active transportation that we are seeing here is example is sodium potassium pump. Usually we see that our excitable cells like neurons, okay, and your cardiac cells which are going to be present, they are excitable cells. And these excitable cells are going to usually have cations like sodium are more high in concentration outside, while to that of compared to that of the potassium ions which are present inside the membrane. Okay, so the sodium ions are more when compared to that of the potassium ions here. So remember that in the active transportation process, what we are doing is we are moving the ions from a lower concentration, children. Let me write this for you. This will be clear for you now, that we are moving from a lower concentration towards a higher concentration, children. So you will be moving from a lower concentration to higher concentration. So this requires, so this is the first point that you need to understand. The second thing is, as you're moving uphill, this is being called as uphill transportation, where you are moving from higher concentration to lower concentration. So obviously we require the ATP. So ATP is being used to move the ions or molecules from the lower concentration to the higher concentration here. Here also we are going to use the membrane proteins are the carrier proteins. We are going to even require the carrier proteins to move these ions or molecules across the membrane. So this is important to remember, children. So let us take one particular case here about the sodium potassium pumps that I told you that here you can see that the one here, these are the molecules which are complementary, right? So what is happening here is the complementary molecules are going to get fixed into this particular carrier protein, okay? But in order to move these across the membrane, now we require ATP, because you know that you can see that the sodium ions are very less, as I told you, inside the membrane when comparatively to that of the extracellular space. So you're having more concentration of sodium outside than the concentration inside. So we are moving the lower concentrated sodium to a higher concentration. So first of all, the complementary sodium, see, all the carrier proteins are very specific to their own molecules and ions. So this carrier protein is specific to the sodium. So they got binded here, they're complementary, they got binded How many of them? The 3 sodiums. Okay. Now what is going to happen is one ATP molecule is going to get binded to this particular carrier protein. And now what happens is this ATP, you can see that the inorganic terminal phosphate will be hydrolyzed. The bond will be broken and it is going to form what? ADP, adenosine diphosphate, and one of the inorganic phosphate molecules is being attached till here. So once the terminal bond is being hydrolyzed here, energy is released. And due to this energy released here, what has happened is there is change in the shape of the carrier protein. Earlier it was opening towards inner side. Now because of the energy, changed its shape and it has released all the 3 sodium ions outside into the extracellular space. You can see the change, children. And once it's been opened up over here, 2 of the potassium ions, which are complementary to the carrier protein, got fixed here. And now what has happened? Once the phosphate molecule is getting released off, once the phosphate is going to be released off, see, the phosphate is leaving off. So now what happens is there is change in the shape of the molecule. Again, now it is going to flip and open towards intracellular membrane and now intracellular side. So now what happens is the potassiums which are being fixed are being released out. So the exchange is between the 3 sodium ions pumped out by the hydrolysis of ATP, and while the shape, there is change in the shape again, as a phosphate molecule is going to be left by the carrier protein, there is a change in the shape. Again, it's going to be flipped back inside the intracellular membrane, and the 2 of the potassiums which are attached here are going to be dropped into the intracellular space. So this is going to be the sodium potassium pump, where you can see that from a lower concentration to higher concentration we are trying to pump. We also know that the potassium ion concentration is very, very less in the extracellular space, but with the help of this particular carrier protein and the hydrolysis of an ATP molecule, by changing the shape of the carrier protein, we could bring the potassium from a lower concentration to the higher concentration here. So these are the basic things that you need to remember. And remember, children, usually the ATP pumps are being seen, that is active transport is being seen usually in your what we call is the sodium potassium pumps we can see, and this can be usually seen even in the co-transportation also, like we have seen in the co-transportation about selective reabsorption in the kidneys. When I made a video regarding that, I have explained you. And the co-transportation is also going to happen in the ileum during the glucose absorption into the bloodstream. Okay, so that is also there, the co-transport. So I'll make a special video on the co-transportation of the glucoses, by which you can understand the transportation of molecules or ions across the membrane very easily. And next, coming on to children, we are going to have another final process, which is going to be the osmosis process, that I need to make you understand. So for that, let me share my screen with you all, come to the concept of this particular osmosis process. It is also one of the easiest processes that you can remember. So let me take up this particular procedure now. I'm speaking about the final procedure, which is going to be the osmosis, children. So osmosis is something where you can see that the water molecules are going to move from a higher water potential to a lower water potential. This is my way of representing a process instead of writing in a sentence form. Okay, so what are you moving is you're moving water molecules. So let me tell you that, for instance, what is water potential? Let me define first what is water potential. Okay, so we all know that your water molecules, basically we have spoken the diffusion process, that our fluids, okay, our fluids and gas molecules are going to usually have their own kinetic energy, right? So because of the kinetic energy, what happens, the molecules will be randomly moving, and what they do is they are going to apply pressure on the wall of the container, right? If this is going to be my container, the molecules of the fluids and gases, because of the kinetic energy and the random movement, what happens is they apply pressure now. So this pressure, the pressure applied here, or the pressure created, what I can say, the pressure created by water molecules, by the water molecules, is being called as water potential, children. This is being called as water potential. So this itself is water potential. water potential. So children, now what I'll do is water potential is being designated as a psi symbol. This is a psi symbol, okay? Usually the pure water, that is only the water, okay? The pure water is nothing but which does not have any solutes, okay? This is going to have its water potential as what, you know, the water potential is going to be, the value is going to be 0. So this is the only positive value that it is going to have. For instance, if I'm going to take a beaker of water, okay, this is my water, children, and if I'm adding some solute into it, I'm adding some sugars into it now, children. So what is happening is my pure solvent was my water. This was my water, but I added solute into this. So now it has become a solution. It has become a solution now. Now what is happening is the solute molecules, now what they'll do is they will decrease the water potential. So the solute molecules, what they are doing is they are decreasing the water potential. They are decreasing the water potential or the kinetic energy of your water molecules. You can just remember this like a traffic on the road. If you're traveling on an what we call is a road, which is vacant, but you can have a very good speed. But if you're going to have multiple vehicles around you, then your speed is going to be reduced. So same as in the way the solute molecules are decreasing the water potential. So if the water potential decreases, now what happens is, children, the water potential is going to move towards which value? Do you know? It is going to move towards a negative value. So if you're adding a solute to the water, what is happening is the water potential is going to be going towards a negative, and the pure water is going to have a greatest water potential, that is a positive value, which is going to be 0. And you can measure the water potential. You can measure the water potentials in kilopascals. We can even measure them as the kilopascals. This is about the water potential. Now let me take two of the things where the water potential towards this side is going to be something like it's going to be a higher concentration, and here the water potentials is very, very less, children. It's going to be the water potential is very less over here. So what we can do here is now, according to our osmosis process, we require a semi-permeable membrane also. So this is going to be your semi-permeable membrane. So if the water molecules, semi-permeable membrane. So the movement of water molecules across a semi-permeable membrane from a high water potential to lower water potentials is being called as what? This process is being called as osmosis process, we are going to call. If you remember, children, in your schools or your colleges, you have learned about a potato osmoscope experiment and a thistle funnel experiment. Let me take a thistle funnel experiment. So if this is going to be your thistle funnel, and your thistle funnel is going to be what you call is tied up with a egg membrane here, which is going to act like a semi-permeable membrane, and this particular beaker is being filled with water, and your thistle funnel is being filled with the sugar solution. So this is going to be sugar added into the water. So as you're adding solute into the water, what happens? Your water potential is going to be reduced, whereas this is going to be only water. So the potential of the water is obviously high. So according to the osmosis process, where the water molecules move, it is going to be from the beaker to the top of the thistle funnel. So after a while, what happens is the level of the water raises here and it stops here because it is maintaining equilibrium. So once they both are going to be that is isotonic, they are going to be isotonic, the water molecules will be going on moving from a beaker to the top of the thistle funnel until and unless it is going to maintain isotonic solution. So this happens in all the processes, whether it might be the diffusion, the facilitated diffusion, or it might be your what you call as your osmosis process. The movement of the ions and molecules are being seen till there is isotonic environment built up. So this is movement of water molecules across the semi-permeable membrane from a higher concentration to lower concentration. So based on this now, children, we will see some conditions like water hypertonic and water hypotonic and isotonic. Okay, I'll speak about the hypotonic. So now let me speak about, I'm considering a cell here. This is my cell, and my cell is being placed inside a hypotonic solution. What is hypotonic solution? Is a solution which is, what is tonic? Tonic is nothing but, let me take another color. So what is a tonic? Is nothing but, tonic speaks about the solute. Solute. What is hypo? Very less concentrated. So it's nothing but solute levels are very less. So solutes are very less, and the amount of the water is more than the solute here, children. So this is my hypotonic solution when compared to that of the cell. Okay, so now cell is what? Cell is actually hypertonic. Okay, cell is hypertonic, is nothing but it is having more concentration of solute and it is having less concentration of water. So my cell is placed in hypotonic solution. It is being placed in a hypotonic solution. So you all know, according to osmosis, that the water has to move from a higher water potential to lower water potential, right? Here the water potential is less because solutes are more. If solutes are more, the water potential is going to become more negative, right? Here the water potentials are little bit positive because of very less solutes present here. So now, children, what happens is the water moves from a higher water potential into lower water potential. So obviously now what happens is the cell which is being present here is going to take up more water and it is going to swell, and it is becoming very turgid, very tight completely. The water is applying pressure here on the membrane, okay, and it is becoming turgid. Now, what happens if I am going to place my cell in a hypertonic solution? So again, here also we can see that something is going to run here. So this is my hypertonic solution. So how we are going to consider this is, hyper is more, tonic is solute. Solute is very high when compared to that of the water in the solution. Now let me take even my cell here. So what are the basic conditions in my cell now when compared to that of the hypertonic solution? This is having a very less solute and my cell is having more water. So basically it is having high water potentials and this is having less water potential. So now what happens? The water potential from high to low, the water has to move. So now what happens? The cell now is going to actually, it's going to be shrivel or it is going to be shrink or crenated. So the cell is going to shrink now. Okay, and we call this as crenated. The cell is getting crenated. So these are the obvious things that we can see that here osmosis is running. Osmosis is running. Children, imagine if your cells are placed in an isotonic solution. Isotonic is nothing but the concentration of the water, the concentration of the solutes, both in the cell as well as these are equal. So there will be no net movement at all. There is no net movement because they both are in isotonic, what you call is a concentration. So when you're placing in a hypertonic solution, then your cell is going to get shrink. And when your cells are being placed in hypotonic solution, they are going to swell or become turgid. These are the conditions and these are the measurement units that you are going to have. Like your kilopascals is the units that we are using for the osmosis. And osmosis requires compulsorily the presence of a semi-permeable membrane, which is being required here. And we all know that if it's a pure water, it has a highest value, that is positive value is going to be 0. And the value when it's going to be solute added, it reduces towards the negative side and it's going to be this negative indication. So that is your osmosis that you can see happening across your cell membrane, children. This we can see the conditions that I spoke about. The hypertonic is when you eat more salty food like you are eating your pickles, chips, or some other sorts of like items which have more salt. So your cells are going to obviously, for instance, if your extracellular fluid is going to be, more having concentration of more solute, your cells may shrink. That is the reason what you feel is you feel more thirsty now. Why are you feeling more thirsty? Because such that you can feed or give up more water inside. Okay, so the extracellular fluids should not be more hypertonic, nor they should not be like hypotonic. It has to be isotonic medias that have to be concentrated. So we have this special homeostasis in our body to maintain all these ion equilibriums inside the body. I hope, children, you all have understood about the different procedures like diffusion, facilitated diffusion, active transport, as well as the osmosis process. So if you like this video, please do hit the like button and try to share my channel with others. Subscribe my channel. Thank you all. Thank you.